Okay, hi everyone. First of all, I want to thank the organizers for inviting me to speak and everybody else for attending so I could share you some of this work that I'm really excited about, um, describing this concurrent inhibition of the RAS pathway and autophagy treatment as a therapeutic approach for pancreatic cancer. So to give a little bit of an outline of what I wanted to talk about today, I wanted to first give a little background into pancreatic cancer and RAS biology, and then talk about Dr. Kirsten Bryant's work as a postdoc in Channing lab with how RAS inhibition increases autophagic dependence in pancreatic cancer, and then how she has set up her lab and my thesis project expanding upon that work, specifically with regard to novel strategies for inhibiting autophagy. And so to put a little bit of this into context, pancreatic cancer is the third leading cause of cancer deaths in the US. With a 13% 5-year survival rate. It has grown over the past few years, but clearly there's still a lot of room to improve upon outcomes for patients. And so what you can see very clearly is that these deadliest cancers, the highest number of cancer deaths in the US, correlate very closely with RAS mutation frequencies, in particular pancreatic ductal adenocarcinoma, PDAC, or just pancreatic cancer for the purpose of this talk. Almost every pancreatic cancer case harbors a KRAS mutation. And so we think about cancer as a multi-step genetic disease, but in particular for pancreatic cancer, these KRAS mutation is the defining event that allows for tumorigenesis and overall progression from this PANN, these early precancerous lesions, to a more severe cancer and metastatic disease. And so what is KRAS? It's a small GTPase that functions as like a molecular on-off switch to regulate critical signaling pathways, in particular metabolism, growth, survival, things of that nature. And mutation of these proteins locks the protein into a constitutive on state so that it hyperactivates the pathway inappropriately and drives too much growth, too much survival, stuff like that. Now, KRAS effector signaling is actually fairly complex. This diagram is supposed to be too much and too small to see because it has over 50 known candidate effectors. But what's really interesting in the context of PDAC is a paper that came out over a decade ago describing how the RAF-ERK MAPK pathway that KRAS signals to, how it can recapitulate this KRAS pancreatic cancer phenotype. And so what I'm showing is if you put a mutationally activated KRAS G12D into a mouse, you get these PANNs, these precancerous lesions. And then when you also have a loss of function mutation of the tumor suppressor P53, then you get full progression from just the PANNs to actually metastatic disease. And that's something that can also be recapitulated if you go one step down from RAS to RAF, or in this case, BRAF V600E mutation. Again, you can recapitulate this full metastatic disease with a BRAF V600E mutation and P53 loss. So the reason I'm showing this is because while KRAS effector signaling can be complex, in the context of pancreatic cancer, what we think is actually happening is that it's fairly simple in the sense that this RAF-MEK-ERK or MAP kinase pathway is doing the majority of the heavy lifting to delineate this inappropriate RAS signaling. Now, this comes with a little bit of an asterisk in that the ERK kinase has over 1,000 substrates. So it's not entirely simple, but we do think of this RAF-MEK-ERK cascade as doing the heavy lifting for RAS signaling. And so there's a number of different strategies to target RAS for cancer treatment. And in particular for this talk, what I'm going to be talking about is direct KRAS treatment, as well as inhibition of metabolism. And we talk about metabolism because of the fact that KRAS signaling greatly alters the metabolic state of pancreatic cancer. In particular, it makes it highly dependent on these nutrient scavenging pathways like macropinocytosis and autophagy, but also with a lot of other critical metabolic pathways like glycolysis. And for the purpose of this talk, what I want to really focus on is autophagy, which is this process of cellular self-eating of the cellular components. And the reason that we as a lab care so much about autophagy is because it's been shown time and time again that autophagy is absolutely essential for pancreatic cancer tumorigenesis, for maintenance, and also to overcome different insults that you put on these cells, whether it's chemo or other therapies. And so with that bit of background out of the way, I wanted to go now first into the autophagy pathway, as well as how RAS inhibition increases autophagy dependence in pancreatic cancer, a discovery that Kirsten Bryant made, again, back in her postdoc with Channing, and how that's laid the foundation for her, not only her lab, but also for my thesis work in graduate school. So when we're talking about the autophagic pathway, we can think about it in 4 distinct phases, with the initiation and the nucleation, which ultimately not only signals the start of autophagy, but also to start building the phagophore, maturation, and then importantly, degradation. And I say importantly because the autophagosome and the lysosome actually fuse at the end of the degradation, and the acidic environment causes the breakdown of these metabolic components into units that the cell can actually use to fuel growth. And I want to stress here that I'm highlighting in yellow this LC3 protein, LC3B. It's a scaffold protein, and it allows for the phagophore to maintain structural integrity, and is built into the growing and maturing autophagosome, and is a way that we can actually monitor flux through the pathway, or just overall levels of autophagy happening inside of our cancer cells. And so we can take that LC3 protein and treat with different inhibitors of the end of the pathway, in particular hydroxychloroquine or chloroquine for the purpose of this part of the talk, which is the only FDA-approved autophagy inhibitor. So the hypothesis was that if autophagy is required for pancreatic cancer tumorigenesis and maintenance, and essentially all pancreatic cancers harbor a KRAS mutation, then it must be very likely that KRAS drives elevated autophagy in these mutant pancreatic cancers, and that loss of KRAS will ultimately impair autophagy. And so to get at this question, we can use that LC3B protein, but in this case, express it in our cancer cells with an EGFP and mCherry tag. And so what happens is you can monitor the autophagosomes as they move through the pathway as being green and red, up until they fuse with the lysosome, and that acidic environment actually quenches the EGFP, and therefore you're left with only the red signal. So you can use an autophagic index of mCherry to EGFP to monitor what's happening to autophagy as you do these different insults. And so what Kirsten initially found as one of her first experiments is the exact opposite of what her hypothesis was, in that KRAS silencing and ERK MAP kinase inhibition actually increases autophagic flux. And so what I'm showing here in the middle graph, you treat either in green with a non-specific siRNA or in red with an siRNA against KRAS. And you can see across our panel of cell lines, as you move from green to red, autophagic index is actually increasing significantly. And that phenotype is also recapitulated if you use a pharmacologic inhibition of ERK. Again, in green with DMSO on the left, or excuse me, on the right side, and then red with the ERK inhibitor, you can see a significant increase in autophagic flux when you silence either the KRAS pathway itself or downstream with ERK inhibition. And so then it seems like the increase in autophagic flux is likely a resistance mechanism to try to overcome this perturbation. And so Kirsten's next question was, does this present a therapeutic vulnerability in that you treat with not only an ERK inhibitor to increase the autophagic dependence, but then on top of that, an autophagy inhibitor to kill the cells? And so indeed, what we're seeing here is if we use our chloroquine, our end of the pathway inhibitor, if you look at these human cell lines in red, you see a dose response of ERK inhibitor as you move from left to right across the x-axis. And then in black, when you add on chloroquine, you can see a nice sensitization of our cell lines to chloroquine, indicating that you're getting a better growth impairment in combination than either drug alone. And again, on the right side with the human organoids, you can see that this sensitization phenotype is very much maintained. Again, making exciting that it seems like if you do treat with an autophagy inhibitor on top of an ERK inhibitor, these cells can't seem to overcome this insult. Moving from 2D and 3D in vitro data, you can actually see this phenotype really, really nicely when you do a mouse model with this very similar experiment. So at the top left there, you can see the controlled tumors grow out as you might expect, and then hydroxychloroquine in green largely doesn't have an effect on proliferation. And the ERK inhibitor in blue does have a significant decrease in proliferation of these tumor cells. But, excuse me, in red, the combination of the ERK inhibitor and hydroxychloroquine, you can see a significant decrease in relative tumor volume. And that's nicely displayed in the bottom and in the middle here with a significant decrease in tumor weight. And what's very exciting too is that the survival is 100% with the combination, indicating it's not only effective at treating these tumors, but the mice are also tolerating this combination very well. And so Kirsten wasn't the only person to find this. Actually, 2 other groups came to the same conclusions independently. And so that ultimately led to a co-submission to Nature Medicine with both Kirsten and Channing as well as Martin McMahon's group. And what was really exciting is when this happened in 2019, it actually sparked a resurgence in autophagy inhibitor interest, which was a very timely thing, as the 2016 Nobel Prize in Medicine was just awarded to the person who actually discovered autophagy and characterized it not only in yeast and mammalian cells. And so what was very, very exciting for Kirsten and Channing is that these studies led to initiation of multiple clinical trials across the country, trying to test the efficacy and safety of MEK-ERK and autophagy inhibition in pancreatic cancer. And in particular, I'm going to talk about the one that our lab was involved in the Dana-Farber and Brian Woolpin, again, testing ERK inhibitor alone and in combination with the autophagy inhibitor hydroxychloroquine. These data were actually just presented a few weeks ago, and unfortunately, it's largely a negative result with this trial. And in particular, one of the biggest drawbacks that they were seeing was that there were significant toxicities associated with ERK inhibitor, which resulted in suboptimal target inhibition. They weren't able to dose it high enough that they think they could fully inhibit the pathway. And we do know that hydroxychloroquine as an autophagy inhibitor suffers from pretty poor PK/PD in patients. And so with the lack of assays to assess on-target activity, it doesn't seem like it was a great autophagy inhibitor in combination with the ERK inhibitor. And so with those somewhat negative clinical trials, the question is, how can we build off of that? And so a lot of my thesis project has been this 2-pronged approach of improving upon the ERK inhibitor arm of the combination, as well as the hydroxychloroquine, right, the autophagy inhibitor arm of the combination. And so with that being said, I'll take us back to the outline, which is going to start with this initial part of the better autophagy inhibitors arm and finding novel strategies for inhibiting autophagy. And so to start off my thesis project, we did this CRISPR screen where we took our cancer cell lines and infected them with a druggable genome library, which is essentially guides against genes that encode proteins that are druggable, whether FDA-approved or preclinical compounds. It's a largely druggable, therefore hopefully more translational findings for this screen. And unsurprisingly, in the vehicle-treated arm of this screen, a lot of these orange dots fall out, which are all autophagy genes. It's something we would expect because, again, autophagy is very critical for pancreatic cancer growth and survival. And in particular, we find PIKfyve as a significant and very essential gene to PDAC proliferation. And we were interested in this not only because it was a lipid kinase, which we'd been studying for a little bit now, but also because there were clinical compounds at the time that were in the clinic against this target. And so what is PIKfyve? So PIKfyve is a lipid kinase that actually phosphorylates PI3P to PI3-5P2. And the purpose of this phosphoinositide product is to allow for the autophagosome and the lysosome after fusion to actually undergo fission and get the recycling of the vesicles back through the pathway so that autophagy can continue to happen in a very cyclical type of fashion. So when you inhibit this protein, you don't allow for this fission process, and you see a huge buildup in these vacuoles throughout the cell. And so the first thing we wanted to do was just simply validate the screen. So again, we used this clinical compound apilimod as our PIK5 inhibitor and titrated in a dose response in our cell lines. And we can see, as you might expect, that it impairs growth in our pancreatic cancer cell lines quite effectively in this 5-day assay. And what we were also wondering is whether it was actually a durable response. So we did a colony formation that was a 14-day assay. And what I'm showing here at the top is an increasing dose of PIK5 inhibitor out to 14 days, and then staining with crystal violet to just simply quantify the colonies. And you can see at 14 days at these higher to mid concentrations, there's almost no outgrowth of the cancer cell lines. And we were excited about this because even compared to a 5-day assay, you can see at the bottom left there, chloroquine has a GI50 in our cancer cell lines closer to the 10 to 20 micromolar range, whereas apilimod or a PIK5 inhibitor is much more in the tens to hundreds of nanomolar range. And again, this is seen very nicely when you compare the two at these 14-day assays. Even at the higher concentrations at the top right of chloroquine at 3 and 6 micromolar, our pancreatic cancer cells are still able to overcome that autophagy inhibition and outgrow, which is not seeming to be the case with PIK5 inhibitors. This is a lesson in looking under the microscope at your cells after you do stuff to them. And one of the really cool phenotypes we saw compared to the DMSO to the PIK5 inhibitor treated are these huge increases in vacuoles that you see. And this has actually been described by other papers using not only this compound, but also genetic approaches to get rid of this protein. And it's a very unique phenotype underneath the microscope compared to chloroquine. And so one of the things that we wanted to do is figure out what these vacuoles were. And so we stained very basically with either this early endosome antigen 1 in green or this lysosome associated membrane protein in red. And what I think you can appreciate very clearly is in these PIK5 inhibitor treated cells, you see a huge enrichment for LAMP1 along the outer membranes of these vacuoles inside the cells, and almost no early endosome staining in the PIK5 inhibitor treated cells, indicating that you're seeing an exhaustion of this early endocytic machinery and everything's getting stuck at the end of the lysosome pathway, unable to undergo fission. And so while this looks cool, our question was, does PIK5 inhibition impair autophagy in our pancreatic cancer cells? And so again, I'm going to take everybody back to this probe that we have. And so what we're expecting to see with this LC3B EGFP mCherry probe is now we're going to inhibit autophagy, prevent acidification of the lysosome, and you should actually see a buildup of green because you're not able to quench the green fluorescent signal. And so indeed, what you can kind of see here, right, when you move from either the DMSO in green to the PIK5 inhibitor and chloroquine in green, is a significant increase in just green puncta, overall green fluorescence, and that's quantified below. When you normalize everything to the untreated DMSO in blue, you can see a significant decrease in autophagy with both the PIK5 inhibitor in red and chloroquine in green. And while you might say that these two drugs look like they're doing the exact same thing, what was really exciting for us is that we can achieve this level of autophagy inhibition with 100 nanomolar of our PIK5 inhibitor compared to 6.25 micromolar with chloroquine. We can do a very similar assay, but with the endogenous protein. So I'll remind everybody again that LC32 gets incorporated into the growing autophagosome. And if you inhibit the lysosome with either apilimod or chloroquine, you should expect to see a buildup in LC3B2 as it's not being degraded. And what we're seeing just via Western blot is at the top, when you increase your doses of PIK5 inhibitor at 24 hours, in all of our different cell lines, you can see a very significant increase in LC3B2 accumulation. And that's not only happening immediately at 24 hours, but also sustained out to 5 days, indicating we're very durably inhibiting this pathway. We wanted to know whether this was actually happening in vivo as well, or if this might have just been an artifact of cell culture. And so one of the first things we did to get at that question is a pretty quick mouse experiment where we took these KPC-derived cell lines, orthotopically implanted them into the pancreas of black 6 mice, and then oral gavaged apilimod over the course of 3 days, and then did a simple tumor extraction to collect some of the tumors for lysates and protein quantification, and then some to send off for histology and H&E staining to look at overall tumor architecture. And a really exciting initial result that I'm showing here with the H&E staining is that when you move from the vehicle treated, the zoomed in pancreatic cancers to the PIK5 inhibitor treated, you can see a significant accumulation of these vacuoles, very, very similar to what we actually see within a 2D cell line in a plastic dish, indicating again, we're inhibiting PIK5 in vivo, and that this treatment concentration and regime is also really nicely correlating with an anti-proliferative phenotype. Again, just doing a Ki67 stain, as you go from all the brown cells in the vehicle to almost no positive Ki67 staining in the PIK5 inhibitor treated, we're seeing that this apilimod is able to actually prevent proliferation within the tumor compartment of these mice. And again, we were very excited to compare the vehicle treated to the PIK5 inhibitor treated tumors. You can see a significant amount of LC3B2 accumulation in the PIK5 inhibitor treated tumors, indicating we're getting not only on target inhibition of PIK5, but it is actually conferring an anti-autophagy phenotype within this living model, which was very exciting for us to see. And so with all of that, we think that we found a very effective, better way to inhibit autophagy to improve upon hydroxychloroquine. And then the next question was, how can we improve upon the additional sensitizers? And I'll remind you that the reason for doing that is because we're seeing from these initial patient trials that this ERK inhibition is a little bit too toxic for the patients to tolerate. And so therefore we need an alternative approach to not only make the cells more addicted to autophagy, but also to spare the healthy tissue within the patients, obviously. And so Many of you who have been in the field know that RAS has been undruggable for nearly 40 years. And a huge breakthrough happened just a few years ago in which we got the first approvals of direct KRAS inhibitors, in particular against a specific mutation of KRAS, KRAS G12C in non-small cell lung cancer. And while this is just a small subset of pancreatic cancers, it's still a huge breakthrough in the field to be able to do this and also get approval. And so what this has allowed for too is a massive explosion of other direct KRAS inhibitors. And so this slide's courtesy of Channing, who very nicely outlines not the inhibitors that are available and their status in terms of clinical trials, preclinicals. And so what you can see is there is a ton of different ways to now inhibit KRAS, both different mutant-specific inhibitors, but in particular, what I want to talk about is this RAS wild type /X. So these broad spectrum pan-RAS inhibitors, in particular, this RMC 7977, a drug from Revolution Medicine. These papers came out a few months ago, and I encourage everybody to read it because it's a huge breakthrough in the field to allow for this tri-complex formation of cyclophilin A and the different mutations of RAS to inhibit effector signaling. And it very potently prevents RAS from signaling. And what's very, very exciting is very recently they showed the first in human trials with this RAS inhibitor as a second-line therapy, and they're seeing outstanding disease control rate of almost 90%, which is something that is typically unheard of for second-line standard of care for pancreatic cancers. Revolution Medicines admits that these results are obviously phenomenal, but they are a little bit short-lived, and so the company and the rest of the field is very focused on combination strategies to try to improve upon and prolong the disease control rate that we're seeing initially in these patients. And so one of the first questions we had is what happens to autophagy in our cancer cell lines when treating with this drug? And so as you might expect from the genetic silencing experiments and the ERK inhibitor experiments that I showed you initially, as you titrate in the RAS inhibitor, you can see a dose-dependent increase in autophagic flux in our panel of cell lines. And so then the question that we had was, can a PIK5 inhibitor on top of this RAS inhibitor actually prevent this induction of autophagic flux? And indeed it does. What I'm showing is everything normalized to blue to the DMSO. And as you treat in red with a RAS inhibitor at 72 hours, you can see a significant increase in autophagic flux. And then when you add a PIK5 inhibitor on top of that RAS inhibitor, you can see a rescue of that induction of flux almost back down to baseline, which is something we were very excited about. While this is cool, the next question, of course, is, does this blockage of induction of flux actually confer an anti-proliferative phenotype in our cancer cell lines? And so the next thing we wanted to do were some proliferation assays to see how the combination works in terms of impairing proliferation in our panel of lines. And so what I'm showing here on the left in blue is a dose response curve of the RAS inhibitor by itself. And then as you step down in different colors, we're adding not only that RAS inhibitor, but also a fixed concentration increasing of the PIK5 inhibitor. And so what you can see is a nice sensitization from right to left of the GI50, indicating that this combination is working very well together. And I'm not showing here, but this combination ends up being very synergistic when you quantify via Bliss synergy. Again, this is a 5-day assay, and so we're curious about whether or not this improves the durability that we've seen. And so going back to the colony formation assay, we can treat with either a fixed dose of a RAS inhibitor, or then in combination with increasing doses of PIK5 inhibitor. And what's very exciting to see at the top right of this is that you see almost no cells, even at these low doses of PIK5 inhibition, indicating that the combo is working very well together and durably inhibiting growth, even out to 2 weeks in our cell assays. And again, this is something that's, I think, really nicely demonstrated in terms of PIK5 inhibition being better than chloroquine, because even with the addition of the RAS inhibitors, chloroquine still doesn't quite outperform PIK5 inhibition in terms of durably impairing growth. Finally, most of this has been done in 2D cell lines, but then we wanted to extend this assay into our 3D models of patient-derived organoids. And we were very excited to see, again, a sensitization from right to left as you increase the doses of PIK5 inhibitor in combination with the RAS inhibitor. And then again, below each curve, these Bliss synergy heat maps indicating in red high levels of synergy between these two drugs, indicating that this could be a potential effective therapy for this particular cancer type. And so then just to kind of conclude and summarize for everybody, what I hope I've convinced you of is that, Kirsten very nicely showed initially that concurrent inhibition of ERK and autophagy can suppress pancreatic cancer tumor growth. Looking to improve upon that hydroxychloroquine as the only FDA-approved autophagy inhibitor, we found that PIK5 inhibition very effectively can block autophagy in pancreatic cancer cells. When you combine PIK5 inhibitors with these new very exciting RAS inhibitors, you can see synergistic reduction in pancreatic cancer proliferation in our panel of 2D cell lines, and that this synergy is maintained, if not even enhanced, when you put them into 3D models of our patient-derived organoids. And then finally, what we were very excited to see is what seems to be on-target PIK5 inhibition activity in our orthotopic models of pancreatic cancer, which also confers LC32 accumulation, indicating that we're able to inhibit PIK5 enough to actually inhibit autophagy in these tumors. And so there's still, of course, more work to be done. Something that's ongoing at the time is performing combination mouse study experiments. So the RAS inhibitor plus PIK5 inhibitor in a longer term mouse study to not only look at the treatment efficacy in terms of changes in tumor volumes over time, but also how well tolerated this combination is. We've gotten some hints from other labs as well as a little bit of clinical trial data that apilimod, this particular PIK5 inhibitor, might suffer from similar poor PK/PD. So we're also aiming to use alternative PIK5 inhibitors before we start trying to transition this to the clinic. All of this work was done in the context of pancreatic cancer, but we'd also like to extend these combination studies to other KRAS-addicted cancers and autophagy-addicted cancers, so things like lung, colorectal, and melanoma. And we also don't think that we're entirely done, and we haven't necessarily maybe found the best autophagy inhibitor. So an ongoing strategy or an ongoing process in the lab is identifying novel strategies to more potently inhibit autophagy in our cancer cell lines. And so then I just want to take a moment to thank Channing Durr, Kirsten Bryant, and Adrian Cox for allowing me into the super group to be able to run around and do these experiments. The whole lab listed has been an enormous help in getting this project off the ground and now in revision at Cancer Research. But in particular, people that are bolded are not only authors, co-authors on the paper, but have also done a lot of work to help me further this and learn how to just do science a little bit better. So with that, I thank everybody for their time and attention, and I'm happy to take questions.